Hello, everyone. This is our presentation on hypoxic ischemic events and their physiological effects on the brain. So first and foremost, we want to understand what hypoxia and ischemia mean. Hypoxia is a condition where there's reduced oxygen supply to the brain, and ischemia refers to a reduced blood flow, which limits both oxygen and glucose or nutrients to the brain. So an overview of brain physiology. The brain relies heavily on oxygen and glucose as its main energy sources. Through aerobic respiration, the brain produces ATP, the energy currency of cells. ATP is critical for maintaining ion gradients, which support neuronal signaling and brain activity. Brain tissue is vulnerable because of its high metabolic demands and limited ability to store oxygen or glucose. Glucose is a critical energy source for both the adult and developing brain. With oxidative metabolism providing most of the energy needed for brain functions, increased neuronal activity raises energy demands, leading to metabolic and vascular adjustments to support enhanced brain function. Proper brain function relies on tightly regulated energy metabolism at various levels, from regions to individual synapses. The neurovascular unit concept highlights the intricate relationships between brain cells and cerebral blood vessels, including neurons, glial cells, and vascular cells. Neurons and astrocytes are central to brain energy metabolism, with neurons primarily using glycolysis and astrocytes relying on oxidative phosphorylation. This triggers a series of secondary intracellular events driven by glutamate and tumor necrosis factor alpha, which ultimately results in cell death through apoptosis and necropoptosis pathways. Simple schematic outlining the pathophysiological responses following traumatic brain injury, or TBI, and the complex outburst of secondary impairments. Note that secondary injury processes of traumatic brain injury include blood-brain barrier disruption, neuroinflammation, excitotoxicity, metabolic impairments, apoptosis, oxidative stress, ischemia, and others. Associated with BBB or blood-brain barrier impairment, microglial and astrocyte activation, leukocyte infiltration, and upregulation of pro-inflammatory cytokines are characteristic of the neuroinflammatory response of a traumatic brain injury. Some consequences of cellular injury. Traumatic brain injury can damage the blood-brain barrier, a critical structure that protects the brain by allowing essential nutrients to pass through while blocking harmful substances. When the blood-brain barrier is impaired after traumatic brain injury, it leads to neuron loss, disrupted brain function, and challenges in recovery. The initial injury from physical trauma causes primary damage, but secondary injury occurs later, worsening the effects. This secondary damage can result in swelling, inflammation, and cell death, often leading to long-term brain disorders such as epilepsy and cognitive impairments.
Greetings, everybody. This is Sofia Penarviso. I'll be talking about diagnostic and monitoring approaches for hypoxic ischemic diagnosis, standard assessments, and laboratory findings. One of the most standardized methods to identify cerebral edema is a CAT scan or CT scan. What professionals are looking for is a decrease of attenuation in basal ganglia. There are two signs: the reversal sign, where white matter is denser than gray matter, and also we have the white cerebellum sign, which identifies necrosis or necrotic tissue. The MRI is utilized to identify the restricted diffusion in the cerebellum and basal ganglia. Cerebral tissue decay is identified by the hyperintensification due to the accumulation of denaturated proteins in the cortical laminal necrotic tissue. Laboratory assessments are also required to confirm the deproteination of brain tissue. Laboratory samples consist of blood biomarkers and cerebrospinal fluid. In this section for hypoxic ischemic events, we will review some of the acute interventions, some biomarkers, and also genetic predispositions and post-treatment care. Temperature to 92.3 Fahrenheit to 94.1 Fahrenheit, at least for 72 hours. We also have ventilation, which possibly mechanical ventilation if required to maintain SpO2 saturations at least 98%, and monitoring must be applied constantly. Biomarkers and predispositions vary depending on age or stage in life. In newborns, usually birth complications are the most common cause. In young adults, usually hypoxic events occur due to traumatic brain injury, drug overdose, cardiac arrest, etc. Middle-aged adults, 40 to 65 years old, usually is by the accumulative lifestyle factors that bring them to that stage due to smoking, high cholesterol, physical inactivity, and obesity. And older adults from 65 years old and older, they are at higher risk due to cardiovascular predispositions, such as atrial fibrillation, diabetes, and atherosclerosis. Additionally, there are genetic predispositions to hypoxic ischemic events, such as genetic precursors, such as NOTCH3 mutations or APOE4 allele. And also we have the contributing conditions of sickle cell anemia and mitochondrial disorders.
So how does the brain recover? There are two types of neuroplasticity. The first being structural plasticity, which involves the brain's physical structure changing due to learning or experience or trauma. So new synapses are formed and old ones are pruned away. The other is functional plasticity, in which the brain shifts functions from damaged areas to undamaged areas after injury or trauma. For example, in cases of stroke and brain injury, other parts of the brain can take over functions originally controlled by the damaged region. The future treatment that's currently still experimental is called hypothermia therapy, in which they aim to reduce the core body temperature to help reduce secondary brain damage caused by processes that occur after the stroke, like inflammation, oxidative stress, and the release of harmful chemicals like excitatory neurotransmitters. which all can cause further injury to neurons after the stroke. Hypothermia therapy can help by lowering the brain's metabolic rate, which helps slow cell death in areas affected by the stroke. It also can inhibit harmful chemical reactions caused by excitatory neurotransmitters, which cause excitotoxicity by slowing the release of neurotransmitters. It also helps to reduce inflammation, which can be caused by inflammatory cytokines, which happen from dying neurons. Um, it also helps to reduce brain swelling, which can occur because of the increased permeability of the blood-brain barrier after a stroke, which leads to higher intracranial pressure. And it can also prevent oxidative stress because it helps to reduce the amount of oxygen radicals which are produced from this hypoxic event. So there have been a lot of different studies that have shown therapeutic interventions that can help to decrease the damage of strokes. But when we test them in trial, they produce negative results. And this is due to a lot of features, mainly because of timing, because it's hard to get people that are having a stroke into the hospital within the time frame they need to get these experimental treatments. It's also due to the fact that people that are having strokes are usually people that are very healthy and doesn't have other underlying factors that complicate treatment. Also, just differences between animals and humans in the way that our morphologies and our brains work in these scenarios.